seminario del Instituto de Biología. Antes de presentar al speaker de hoy, quisiera recordarles las medidas de seguridad que vamos a tomar en caso de que tengamos que evacuar el auditorio. Vamos a evacuar el auditorio por ambas puertas y dividimos el auditorio por la mitad. Las personas sentadas en la mitad izquierda van a evacuar por la puerta izquierda y las personas sentadas en la mitad derecha evacuarán el auditorio por la puerta derecha. Los pasillos deben quedar despejados, no se permiten mochilas o cables de celular, nada de esto. Y por último, les recuerdo que ajusten sus celulares en modo de silencio. Hoy tenemos como invitado al Dr. Joseph Eras, quien se desempeña actualmente como especialista asistente en el Departamento de Ecología y Biología Evolutiva en la Universidad de California, Irvine. El Dr. Eras obtuvo sus estudios de pregrado en la Universidad de California, Riverside, una maestría en la Universidad de California, Los Ángeles y un doctorado en la Universidad de California, Merced. El doctor Eras está interesado en identificar los patrones de adaptaciones evolutivas en peces marinos, usando técnicas de genética moderna y secuenciación de nueva generación para obtener datos genómicos y de transcriptomas. El doctor Eras tiene múltiples publicaciones en revistas internacionales, tales como Philosophical Transactions of the Royal Society B, Marine Genomics y BMC Genomics. Queremos también agradecer al posgrado en Ciencias Biológicas, quienes apoyaron la visita del Dr. Eras por medio del fondo PAEC. Entonces, sin más preámbulos, los dejo con ustedes con el Dr. Joseph Eras.
Gracias. Mi español es más o menos, mi presentación es solamente en inglés y si tienes preguntas, dígame por favor. Y también gracias por la invitación aquí, Una. And I'll start my presentation. So yeah, I work on the intertidal and subtidal habitats along the Pacific. So as you can see in this image here, and I have this cartoon of fishes, you know, within the Pacific, and of course I kind of highlighted two kind of groups here. So I have here what are rockfishes. There's about 105 different species of rockfishes, and so they're a really good example of adaptive radiations in marine systems. And so I'll be talking about rockfishes from my PhD work, and then I'll talk about these guys here, which are called pricklebacks. They look like eels, but they're not true eels, and a lot associated with digestive physiology. And so I'll start with just kind of an overview of the questions or the ideas that I'm interested in. So I have this kind of this bottom-up approach. So there's not so much information that I know about rockfishes other than there's a lot of them. There's a lot of species represented, and so I'm taking this bottom-up approach. Whereas there's a lot of information that I know about pricklebacks. So there's, in terms of their digestive physiology, and so a lot of work was done in the postdoc lab at UC Irvine, and so I'm using that information to kind of guide my genetic studies. So as I use the genome of the monkey-faced prickleback to better understand dietary adaptations. And of course, you know, with this we can go further and look at either expression or test of molecular function. And so, you know, with expression we can either look at, you know, qPCR, look at levels of expression, or in situ hybridization, so we can localize where this expression is occurring within a tissue region within the organism. And of course, you know, with RNA-seq, we can do experimental trials to see what happens when we see, you know, upregulated or downregulated genes. And this test of molecular function nowadays that we can knock out or knock down a gene, so lower the expression levels. And it's in these exciting times that we can use CRISPR-Cas9 to manipulate or change, you know, particular locus and see what happens, you know, in terms of fitness for an organism. Right? So, and of course, these are questions that revolve around evolution. So my first question, or my first major project, is looking at rockfishes, right? So as you can already tell, there's so much coloration, a lot of bending patterns, and as you can tell just from the name, rockfishes, they kind of, you know, they look like a rock. So they kind of blend into their habitats. They occupy various depths, and also too, like I had said, that, you know, there's about 105 different species. So my general question is, what are the genes that contributed to this adaptive radiation? So I had mentioned a bottom-up approach, right? So here I have like limited information that I'm going up to use the genetics, right, to look at the transcriptome of these rockfishes, right? And looking at specific tissues, these proteins here, and compare them, and hopefully that I can see these differences at the genetic level that can better explain something about tissue function or about the overall, adaptive trait of that particular organism or that particular fish. All right, so where do rockfishes live? All right, so they're predominantly here in North America, so down here in Baja, through California, up into Alaska. And of course, we also find certain species in Asia as well. So you can kind of see this heat map, right, the coloration. So these species here, mostly reside here in North America. And then the purple colors, you find predominantly in Asia. But the ones I'm focusing on are from North America. And we're only looking at 5 of those 105 different species. So these 5 species here. Also to note, there's about like 22 subgenera within the genus Sebaceus, which are rockfishes. And so I'm only talking about 2 subgenera within the genus Sebaceus. So also another thing that I didn't mention is that some species can live up to 200 years. So that's very long. And of course, they've identified this using the otoliths, the ear bones in rockfishes. So this was taken about, back in 2013, and this is a short raker rockfish. And so these are very interesting questions to look at, especially from a genomics or transcriptomics perspective of trying to find loci associated with lifespan. All right. The other thing that's very interesting, or what has been done, is associated with depth. So Travis Ingram in 2011, he constructed this type of model. So if you can see on the right side, these phylogenetic trees, his model is where if you see the short branches, that character contributed to the speciation process. And as you can see the long branches in this tree here, that trait didn't really contribute to the speciation process. So he looked at various traits, and so the ones that kind of start to stick out are associated with depth. So like this eye width and also body depth has kind of contributed to the speciation process, and also this depth habitat as well. So this is kind of a key indicator of like probably some type of loci associated with depth would be interesting to look at or investigate further. So here are the 5 rockfishes that I mentioned in my study. So here, as you can see, they occupy different depths, right? Different depth range. And so the common names here go for China, quillback, blue, and olive rockfishes. And so what I did was estimated positive selection. So by looking at the genes, or particularly in the transcriptome. And so positive selection, what we can do is that you have your population, right? And if you have a mutation that's advantageous, right? That locus or loci would be passed on to the next generation, right? And if something that was deleterious or not favorable would not be passed on, and you wouldn't see it in the following generation. And so the way we estimate this is called, KA/KS. So here I have an example. So here we have the nucleotide sequences. Here's the protein sequences. And as you can see in the blue, where we see a change, this would actually change the amino acid sequence. Whereas if you see in the red, there is a change, but it doesn't change anything for the amino acid sequence. So there's this ratio of a KA/KS. So if you see more changes where they're non-synonymous, that there would be a change to the amino acid sequence as opposed to the ones that don't have a change. That's an indication of positive selection. So I set up to test these 2 hypotheses. So here I have, if we have maximum age and depth, right? And these 2 live up to 90, 80 years in comparison to the other 3, right? So I've set to test longevity and also set to test depth, right? So these are considered more shallow rockfish species as compared to Sebastisomus, which are found at lower depths. So this is the flow chart of my methodology. Go out on fishing boats and collect our rockfishes, extract RNA, and then we prepped them and sequenced them at the Vincent J. Coates laboratory at UC Berkeley. And get the reads back, and what I did, instead of looking at gene expression, I was more interested in the orthologs, so I'll explain in a bit, and estimating this Ka/Ks metric that I had described earlier. So identify the orthologs, align them, annotate them, and estimate selection. So just to kind of give an explanation of orthologs, it's the same gene found but in a different species. So if we have our ancestral rockfish that has, the circle represents a gene, we can have paralogs where there's a gene duplication event. So here we have this fish that has the circle and the triangle, but this, species doesn't. And of course, any lineage further than that would retain it, right? And then here we have the blue circle that did not have that gene duplication event, and we can start to assign, orthologs. Again, kind of emphasizing the same gene, but found in different species. All right, so what I did was compare from all 5 of these species, and this is from brain tissue. The reason why I use brain tissue is the fact that it gives a lot of transcripts. Currently, there is no genome available for the rockfish, or for the genus Sebastes. And so I was able to find about 10,000 to 15,000 different ortholog pairs. So these are pairs between each species. And if you notice in the yellow, there's an elevated amount, about 14,000, 15,000. And that's because these are closely related species, or these are the ones that are from Pteropodus, and they're closely related. But if I wanted to look at orthologs across all 5 species, right, it whittles down to 3,800. So across all 5 species here. So with those orthologs from all 5 species, right, here I said to test out for molecular evolution for positive selection. And so here I use a branch sites model. So my null hypothesis is that there's no selection that's operating, and the alternative is how the selection is operating on these branches here. So here's Kovac in China, and again, a reminder, these are the rockfishes that live a little longer than the other 3 species, right? And by the genes that I got back, right, by using a chi-square distribution, I get 274 with a value of 0.05, and then of course a little less with 0.01. All right, so instead of showing you, 274 genes, right, I'm able to kind of whittle that down, and I have it summarized by using the gene ontologies. Gene ontologies is like a description, like a hashtag description of that particular gene, right? It gives some type of information about it, right? And so it's kind of broken down into these 3 different domains. So biological process, so something defining about the biology of that particular gene. Molecular function, so what is it doing, in terms of from a chemical perspective, and of course, cellular component, right? So where is it located within the cell? And so there's this hierarchy. So this is an example that I'm giving for hemoglobin, and so this is again just a description. So these are the results. As you can see, all of these genes they're found under positive selection. As you can see, these have a higher Ka/Ks value, and so these are extremely high where there's only non-synonymous substitutions as opposed to synonymous. Right, so we see oxygen transport being represented here, calcium transport, and then cell surface receptor signaling pathway. And so just to highlight on the cell surface receptor signaling pathway, what that particular gene is MHC, right, which is related to immune function. And so particularly, and this is very common, you see a lot in vertebrates, that this gene's under selection, especially with the alpha chain, and this is where the peptide binding region is. So elicit to some type of immune response. And so what's interesting is that I found other genes under selection that are related to immune function. And so in this study that I did, which was published this year, I've seen also genes, including another type of MHC in previous publications, right? So in 2015 and 2011, that are associated with immune function. And so this may be because of immune senescence. So in the fact that as the older that we get, our immune function starts dwindling, right? Then that leads to mortality. But if you have an improved or very well immune system, right, then you can survive or last longer, right, or live longer in years. So moving on from longevity to depth, right? So again, the same premise where I'm looking at. So here, this clade in particular, these are more shallow rockfishes as compared to these two here. Right? And I found a lot more genes represented, so 543. And so what I found from this data set is that some that are associated with apoptosis, some with development, right here, this morphogenesis, and again, oxygen transport starts popping up again, right? And so this is something that surprised me, or just kind of, I looked in further, right? And of course, this was hemoglobin, and which made a lot of sense. Being the fact that we see a lot of changes with hemoglobin, especially for oxygen transport, being the fact that they occupy different depths. And so I looked at, the subunit alpha, right? So we see like a Ka/Ks value of 999. That's just indicating that there was no synonyms substitutions throughout that whole gene comparing those 5 species. If I was to look at subunit beta, right? We see more purifying selection. We don't see that many changes, non-synonymous changes there. And so what was nice, yes, I had mentioned that there wasn't a genome, but there's one that's roughly out there, so I was able to use the tiger and the flag rockfish genomes and pull out hemoglobin genes from there. And I found that there was selection there as well. And just to kind of give you an idea, these species live a lot deeper in comparisons to the 5 that I was looking at. If we were to add them into my analyses, right, so these are just amino acid, not the entire sequence, but these are the sites that are popping up under selection, right? And the ones in white, I'm highlighting that are unique for that particular species. When we construct a phylogenetic tree, there's also other hemoglobin genes represented that are also under selection, but I did not catch in my initial analyses. So here's the original one. This is the study that I have here. I've included the tiger and flag rockfishes here. Still getting a high Ka/Ks value. Here I found some additional hemoglobin genes unique to these 2 species. Being the fact that I use a transcriptome, maybe they were not represented. And I had this additional clade here. And the reason my analysis did not pick up was because one of the orthologs was not represented. So China rockfish was not found in that analysis. But yet I still see selection there. All right, and again, to remind you, this is only 5 out of 105 different species. So it would be interesting to look at more species to see if this pattern holds up. And also too, what had happened within the past couple of years, there was a paper published recently in 2017 that looked at cod, or gadids, and they've noticed a pattern of their hemoglobin genes being under selection and also associated with depth as well. And you see a lot of these additional genes that are represented in the cod genome in comparison to some of these other species here. So of course, where do I go from here is to sequence more species, right, to identify those hemoglobin genes from 105 different species, and of course kind of match it with the physiology, right? What is it about within their hemoglobin, in terms of like pH or temperature sensitivity, also to oxygen dissociation with hemoglobin. And so by looking at these physiological factors in connection with the genetics, right, we can better explain, with those depth adaptations, right? Why we would find certain species occupying a certain depth in comparisons to others. right? And so that wraps up this first project, right? So I'm going to move on to the monkey-faced prickleback, right? So species, the name is, or scientific name is Seminarios del IB. And so here I'm more interested in what are the genes that contribute to dietary diversity or specializations. But with this system or model system, I come from the perspective, or the lab that I'm working at the University of California, Irvine, is that we know a lot about their gut. We know a lot about digestive physiology of these species, or pricklebacks in general. And so we kind of take more of a top-down approach by focusing more on the gut as compared to other tissues within the body. And so I know what type of genes to look at, digestive genes and genes associated with metabolism. And that's where I've been focusing my studies at UC Irvine. So these are the habitats where pricklebacks live. They live in the intertidal zone, so these tide pools. And so there is a lot of vegetation, there's a lot of algae, various species, a lot of invertebrates that occupy these intertidal zones. And what's very interesting about pricklebacks is that they can live outside of water, still a bit moist, but live outside of water for up to 30 hours. And so here's a phylogenetic tree of some of the prickleback species. And the ones where I have an illustration are the ones that I've been looking at in a lot more detail. And these are the intertidal pricklebacks. And the one I have highlighted in blue, I've sequenced the genome of this species, the monkey-faced prickleback, or Seminarios del IB. And if you'll notice in my talk, or the rest of the talk, I'll label carnivory as red, herbivory in blue, and omnivory in purple. And you can see that, especially with the ones with the illustrations, that we see dietary diversity. We even see convergent evolution of herbivory. So we have Seminarios del IB as an herbivore. Zyphistera mucosus is also an herbivore as well. So the methodology for sequencing the genome, I took whole body skin and muscle tissue and sequenced it using Illumina and PacBio and did a hybrid assembly. And I paired that with Illumina sequencing with the transcriptomes. So I took 9 tissues from our monkey-faced prickleback and constructed RNA-seq libraries. And just to kind of give you an idea, right, of pairing the two, of the transcriptome with the genome, think of the genome as kind of a blank map, right? Here I'm using the United States. What's very useful or helpful for using the transcriptome is now that I have those transcripts, we can start to map them back onto the genome. So I know what those genes represent, right? So now I know like points, on the cities, on my blank map here. And so just to kind of give you some of the data that we get back, or even the differences between PacBio and Illumina sequence data, right? So here's just the amount in megabases, so 29,000 and 84,000 megabases. With PacBio, we use what are called smart cells, right? So 40 smart cells were used. And of course, the pros and cons of using the 2 different sequencing technologies is that PacBio, you tend to kind of get some errors that are being called for certain bases, whereas that error rate is a lot smaller for Illumina. And so that's why we went with this hybrid assembly. So also too, in genomics, measuring N50, right, is a good metric of trying to understand how good or how well that assembly of your genome. Right? So just to kind of give you an idea, with zebrafish, if you were to look at the chromosome lengths, right? So some of those chromosomes are 32 to 76 megabases, right? And here I have my little cartoon fish, right? Let's say we had a genome assembly, and it doesn't quite represent, full chromosomes. We get a lot of fragments, right? And so an N50 is kind of skewed towards those larger fragments, right? And then of course it's 50% of that total, contigs or fragments that we have. So here the total is 30. So half of that is 15, right? And that longest contig or fragment is 5 megabases, right? And again, this is just a good way of trying to measure your genome assembly. So for the monkey-faced prickleback, here I have Illumina-only sequences, and we use a program called Flatness. And so here, if we're to measure like the N50, right, it's pretty small, less than a megabase. And as you can see, by incorporating both PacBio and Illumina, and this is an increase with the SMART cells sequence information, we get to an N50 of 6.67 megabases. All right, and just to put that in context, all right, and this was with last year, that if we were to look at any fish genomes available out there, look at their contig N50, that we place pretty well. That here at 6.67, southern platyfish has about 9 megabase N50, and then eastern happy is a type of cichlid. But this is something that's still being developed, of course, with more improved sequencing technology, that we can get better drafts of these genomes. Okay, so now that I have the genome sequence, right, there's some interesting things that we still want to learn about what's inside our monkey-faced crickleback, right? Being the fact that it's an herbivore, want to know what's going on in terms of digestive enzymes, so carbohydrates, that breaking down carbohydrates into simple sugars like maltose or glucose, lipases, breaking down fats down to glycerol and fatty acids, and then protein, broken down by these proteases into amino acids. All right, so that's what I searched for throughout the monkey-faced crickleback genome. All right, so let's focus on carbohydrates, and particularly amylase. Right, amylase breaks down starches into maltose and glucose. And just to kind of give you a reference, we've measured enzyme activity. So here we have on the right, so this amylase activity. And so you can see that's fairly high in comparison to these other species, right? And of course you would kind of expect, especially from like maybe an omnivore or a carnivore, that their amylase activity would be low because they're not consuming a lot of starch. So here, by using the transcriptome, I measured expression, gene expression. So here, I've only kind of highlighted 5 out of the 9 tissues that I looked at. So the liver, pyloric ceca, which is like a pancreas for the fish. This is proximal intestine, middle intestine, and spleen. And as you can see with the heat map, right, so we have high expression here in the yellow, and then more purple is like downregulated, lower expression. All right, and so these are the genes or these digestive enzymes, the carbohydrates that I've identified. But I'll be focusing mostly on amylase. And what's really interesting that we see is that there's 3 copies. So we have what we call AME2B and then 2 AME2As. And of course our monkey-faced crickleback's an herbivore. And if we were to compare it to some of these other species that we see maybe 2 or 1 copies. And of course they have different diets. And just to kind of look at more fine-tune, we see like a transposable element that's wedged right here in our AME2B. Also some, these hypothetical proteins that are wedged in between AME2A. I also did, again, this like Ka, Ks metric, but I did using a different program called DataMonkey, in which it can visualize that positive selection. So here you see this thick branch for AME2B, right? And again, this is an indicator that this gene is under positive selection in comparison in respect to these other species here, or the genes that are represented from each of the species. All right, now focusing on lipases. And so what's interesting here, and again, this is just enzymatic activity. If you notice, this was published from my postdoc advisor, and you can see that lipase activity is pretty high for the herbivores and also this omnivore here, and which is kind of interesting to think that, is there a lot of lipids in your algal diet, right? If they're consuming a lot of starch or they're consuming algae, is there a lot of lipids, as compared to a carnivorous diet? And so, of course, I set out to look for lipases throughout the genome and use the transcriptome to at least look at expressions and seeing where they're expressed within the different tissue types. And one particular gene family that we focus on is carboxyl ester lipase. And so you'll see that represented kind of here, throughout my heat map. And so what I found was that we found 4 copies of this carboxyl ester lipase represented, right? And so as you can see, this one's a little bit darker green compared to these other 3. And that's because this one, when we look at the sequence alignment, is very different compared to these other 3. And of course, then when we start to compare them with these other model species genomes, that there's only 2 copies represented. All right, and I did kind of a search for selection as well. I did not see anything with using a branch sites test. But if I use only a sites test, so it's only looking at the amino acid sequences and not using a phylogenetic context, I do find measures of selection there. And so this is something interesting that I'm trying to figure out why would there be 4 copies of carboxyl ester lipase, and that may contribute to this fish being an herbivore in comparison to some of these other species. Also, to pair with this, as you can see in my illustration here, within the distal intestine we see a lot of short chain fatty acid production. And of course, with the liver we'll see, lipid metabolism. And I'll show you how I'm comparing these two in the sense that when we dissect our monkey face prickleback and we look at the distal intestine, we see that there's a bulge there. And we know that there's microbial activity, right? And we've done this test for short chain fatty acid synthesis or production. And so here's our monkey face prickleback. Here's Cyphistria mucosus, another herbivore. Cyphistria atropurpureus, which is an omnivore, and Anaplarcus progressans is our carnivore. And as you can see, Cividales is the highest out of these 4 here. Right? And so you're getting these short chain fatty acids, acetate, propionate, butyrate, right, in that distal intestine. So we know that there is some type of fermentation that's going on in the distal intestine, right? So, but how's this being incorporated into the monkey face prickleback? What is the benefits that the monkey face prickleback gets back? All right. So if we're to look at a metabolic pathway, right? And so here we have acetate that would enter this pathway, and if you can see the ones that are in blue, these are all are expressed in my monkey face prickleback. So we know that we do see these enzymes that are functional, right? They're operating. Now, of course, we haven't quite tested, right? If we're to take an absence of acetate or remove the microbiome from their distal intestine, would these pathways just be automatically shut off? This is something that we are really interested in pursuing further. All right, so here's that heat map of those enzymes part of this ketotic degrading pathways. Right? So you can see that they are expressed to some form, to some degree. You see a lot, especially within the liver, and it's still represented in the gut as well. Okay, so now last, talking about protein or proteases, right? So, degrading down or breaking them down into amino acids. All right, so here again, looking at different proteases throughout the genome, and I use the transcriptome to help me out. And so we have all these different proteases that I've identified, right? That of course they're being expressed within the gut, right? As you can see right here with aminopeptidase being highly expressed within the gut, right? So again, our pyloric proximal intestine, and middle intestine. And if we're to look at enzymatic activity, interesting enough, we do see some high activity for our herbivores here, right, and also our omnivore here that's closely related to our herbivore here. So we see this high activity for aminopeptidase. So when I started to look through the genome for aminopeptidase genes, there was quite a bit, right? So as you can see, there's a lot. There's these 2 panels here. So here's aminopeptidase represented, so I have 3 here, right? And then I have another 2 found in a different region of the genome, right? And then of course I was trying to find the orthologs, the syntenic regions in other species, right? In this is stickleback, medaka, and zebrafish. And I found 2 or 3 aminopeptidases. Now, if we were to look at them across all vertebrates, right, and so if we're here, I don't expect you to read this, phylogenetic tree here, but just to kind of give you context, right, so here we have, rodents or humans, dogs, cow, and a chicken, and throughout most of, mammals and birds and reptiles, you'll mostly see 1 or 2 aminopeptidase genes represented in the genome. Now, if we were to go down into fishes, or especially teleost fishes, we see, upward bounds of 2, 4, 3 represented, and our monkey-faced crickleback had 5, aminopeptidase genes. So, in summary, right, that we see selection, right? So we see selection for AME2B for our carbohydrases, for amylase. In terms of our lipases, I did see selection for cell 1, and that was, again, the sites-only test that I did performed, and we saw 4 copies. And here with aminopeptidase, I saw 5 copies. And what has been supported, or this hypothesis, that there's this what's called a fish-specific genome duplication, so that you see multiple copies of genes that are represented in fish genomes in comparisons to other vertebrates. So, I mean, by even looking at that phylogenetic tree and seeing that there's only 1 or 2 copies represented in mammals, birds, and reptiles in comparisons to fishes in general, that this sure that we see probably these genes were probably represented well in the fish-specific genome duplication. And of course, there's 5 copies, so it's also too trying to figure out what is the function of all 5 of these copies. Why would our monkey-faced crickleback need all 5 of these, and to what degree are they operating? Just to kind of give you some little bit more information, so I just completed my postdoc work at the University of California, Irvine. I'll be starting a second postdoc at UC Berkeley, so starting January 1st of 2020, and I've already started working, doing some preliminary work there. And so I'm going to start working on pupfishes, so Cyprinodon pupfish species, and so these are the ones from San Salvador in the Bahamas, right? But what we've been doing is rearing them in the lab, and we already have F2s. And feeding them just a very generic, like a cichlid diet. But in the wild, what's interesting about these species is, so this one here consumes detritus and algae. This other species consumes snails. Not this one in particular, but it consumes mollusks. And then this third species actually feeds scales off other pupfishes or other species. And so what I'm doing with this new postdoc is looking at the microbiome and seeing how different it is, or how similar it is across these 3 species. And of course, I'm just starting with the 16S composition, so seeing what type of microbes are being represented in the gut of these 3 species of pupfishes from San Salvador. And I'll let tech knowledge, so this is from my Ph.D. group at the University of California, Merced. My PI, Andy Aguilar, postdoc Enrico Ruiz, and then 2 grad students, my colleagues. I'd like to thank them, and then also to the group at UC Irvine. So this is the German lab, and of course our collaborators on the projects that I've worked on. And with that, I'll take any questions.
Muchas gracias. Thank you very much for your talk. It was very amazing. And I'm very happy that you are here, that we can discuss some projects in more detail. But what I'm really interested is in your first project associated with the rocketfish. In particularly, have you been looking for optic or eye-related genes, since I suppose that the depth and the transparency of the water could be very different between the most.
Yeah, no, so that's a very good question. Being the fact that I used the brain transcriptome, I wasn't going to get any opsin genes represented. But there has been one study, and it was just a candidate gene, so they only looked at opsin genes, and they did find selection, and so a pattern where it related to depth. And surprisingly, no one has looked at it further. So I imagine there's not just one opsin gene. There's probably maybe more genes related to vision and depth.
Yeah, actually, I was interested in the optic lobe in the brain, that is related with the vision function inside of the brain. So not only opsins, but other genes that could be related
to vision. Yeah, so what I've been based since by looking at all these genes, right? What's kind of helped me or guided me is the gene ontologies, right? So those descriptions of the genes, and I didn't see anything associated with vision that really popped out. But maybe, yeah, if I was to look back to see if that was represented, or even, sure, expressed, but I don't know if there was any, like, high, Ka, Ks values that really stuck out.
Super. And another question that I had was, once that you are comparing these duplications in carboxyl ester,
so the carboxyl ester ligase. Yeah.
And I was wondering, because you compare them with Daniel or other groups, but have you maybe been interested to compare this particular duplication with other sister species that have carnivore habits? Or do you have them?
So let's see. So as these other species here, right? At the moment, I just extracted the DNA for genomic analyses, and so we're barely. And this is something that I kind of helped, but I don't know how we're going to carry the next part because I'll be at Berkeley. But my postdoc advisor will have these genomes available, and of course that's like the first thing that we'll be doing is comparing those digestive enzymes, just those satenic regions with these. For sure, we'll do this species, Cyphistramycosis, Cyphistria atropurpureus, Phyticles chirus, and Enopartus purpurescens. So yeah. So it's coming soon.
Okay, super. Okay, thank you. So, thank you.
Yeah.
Well, thank you, Joe, for coming to Mexico. And I have a couple of questions. The first one is about, well, actually this topic, they have different feeding habits. So with respect to find or identify those genes that make, for instance, difference in the skeleton, in the jaw, to find differences between these species?
Yeah, so that's a very good question and something that I've thought about in the sense that the mouth morphology. That what's interesting with the pupfishes, the PI we'll be working with, it specializes on mouth morphology associated with diet. So like with the pupfishes, they've already kind of identified certain loci for eating on scales or mollusk and detritus. Our lab at Irvine, we haven't gone. I guess that isn't our background or specialty to look at the mouth morphology of these species, especially even though they have the different diets. But I've spoken to my postdoc advisor. I don't know if he has any interest in collaborating with someone to looking at those morphologies, right? To see how they specialize on herbivory or carnivory or omnivory. It's just been focusing on guts.
And the second question is regarding depths. A little bit like what Patty was saying. I mean, when you go to different depths, the perception of color is different, right?
Yes, correct.
And I can imagine that the diversity of pigmentation is different. And whether you see something related to that, to the diversity of color within the depths. And the second question is that it's something curiosity. Why fish, when it comes out in the fish market, it's all pale and
That's a good question. All right. So with the first one, so to give you the citation for the opsins, so Siva Sundar and Palumbi have published this paper on opsins in rockfishes. So that's the one that they've looked at it from that context. And sure, with the coloration, right, that you can, some light visibility, and you look at the color spectrum, that you see certain rockfishes, that there's different colors that are being more represented, right? I think, especially with those lower depths, that I think you're getting more brighter colors. They have internal fertilization, so they have to find their mates, right? And then they also have to probably distinguish between, what species, from their own species as comparisons to another species. So I'm sure there's also probably some type of selection. That's something I haven't looked at or I've been curious about, is genes associated with pigmentation. So the pigments that they produce so that that they're being recognized by their counterparts. And your last question about the coloration loss, that is something that I would say transcends all fish species, like especially when you look at museum collections or even when you try to preserve them, that the dissections that I've done, that they start losing their color really quickly. And I don't know if that's probably associated with stress, or just now that these proteins or genes are no longer functioning. But I haven't looked at that. Yeah.
Thank you for your talk. What tissue do you use for the transcriptomic analysis? And second, do you have some estimations of the variability of the expression among individuals of the same species?
So you're talking about the rockfishes, or which studies? So your first question was which programs I used?
No, tejido.
Which tissues for? So for the rockfishes, this was just brain tissue, only brain tissue. And then for the pricklebacks, I used 9 tissues. And that was just, sure, it characterized the genome. So we used brain, heart, gill, pyloric ceca, middle intestine, proximal intestine, and liver and spleen. So those, I had that to just characterize the genome, to see what, to just have all the genes that were expressed or a broad range. But then, of course, as you can see in those heat maps, right, I'm only focusing on the ones associated with digestion. And then, so what was your second question again?
Do you have some estimation of the variability of the level of expression among individuals of the same species?
Yeah, so this was more of a characterization of the genome. So I know that a lot of gene expression studies will use multiple individuals to look at the variants. So this is only where I have like 1 individual or 2 individuals to represent that tissue type. But yeah, and in the lab, we've done other more experimental studies where we've actually taken, so this is work that's been done in the lab that helped, that I hope will be published, by next year. Let's see. So we have a graduate student in the lab that's working on these other species, and we've done these experimental trials where we've given them a carnivorous diet. So even though they're an omnivore and herbivore, that yes, they'll still consume a carnivorous diet. So we'll give them predominantly high protein. We'll feed them flatfish and supplement it with some lipids and other nutrients. And we have like 3 or 4 individuals for each trial. So 3 or 4 individuals per species, right? And so of course we can look at the variation that occurs, in our experimental trials. And this was like a 4-week feeding trial that was done on these fishes.
Thank you for your talk, Joseph. It was really cool to see all this multi-genomic stuff. I have also 2 questions that are probably 10. The first one is, I was wondering, both of them have to do with the sampling of the dishes you did. So the first one, rockfishes, was brain.
Yes.
I was wondering if you were harvesting the brains from individuals you had in the lab and you had control conditions, so you were sampling them from the medium and if you sampled them at the same time intervals, or if you had some that were from late afternoon, early morning, night, or?
Yeah, so they're all wild caught. Yeah, we weren't keeping them in our or rearing them or keeping them in captivity. So so there probably is some type of stress response, being the fact that we're hook and lining them. So we're reeling them up, and then I go in and dissect out the brain and then preserve it in RNA later. And so yeah, and I only use one individual to represent the transcriptome for that particular species.
Yeah, but you did that at the exact same time, or well, similar time, or did you do them during various times of the day?
It was various times, and so that's a very good question. And so even when we published that paper, there was that question, and I think our justification or in the fact that we're looking at the transcripts. So just these are just the mutations in the transcript, and not looking at expression levels. So I could totally see where if I was to do this at different depths or different so many variables that would go on and look at expression. If you look at expression levels, that there would be so many environmental variables that would change that wouldn't be such a great study. And here we're just only looking at the mutation rates. I'm sure these genes are expressed; they're all represented in the fish, but there's those mutations that we're focusing on.
I was just wondering if, for example, on the first set of genes you mentioned, you said that they were related to longevity and AIMHC, but I was wondering if there could be a factor of maybe circadian rhythms, if they were close to night or early in the morning, that could also help explain some of the patterns. I was just curious if that could be, that's why I was asking. And the second question is also related to the sampling and how you dissected the gut of the fish and how you extracted RNA from them.
Yes, so with the pricklebacks, what we do is we go out. So these are all intertidal species. So we go out at low tide. We go with the aquarium dip nets, and so they probably about like 200 millimeters in length, and the adults can kind of, depending on the species, can go up to maybe like 700, like 600, 700 millimeters. But we go in, and this is probably, as of recently, I'm saying this in the sense that I've been extracting DNA for genomic sequencing, and we preserve them in liquid nitrogen. And we also had samples that were just put on dry ice. And there is a very clear difference in the genome extractions with the ones that we did with liquid nitrogen compared to just preserving them in dry ice. The ones in liquid nitrogen do a lot better. And then also too, what's nice is that there's this UC reserves are not too far from the collecting site. So we go collect them, we put them in coolers, bring them back to the reserve, and then we do the extractions there.
I was wondering because I've seen a lot of microbiome studies, and they're cutting the gut into foregut, middle, blah, blah, blah. But a lot of these fish have parasites, and I don't know if when you're cutting the gut, you're opening and seeing if there's not another animal in there, or if you're just cutting it, freezing it, extracting it, and then some of these genes you're finding might not be hosts, but they might be parasite genes, and maybe that's why you see all this other stuff that I wonder about with not just yours, but a lot of microbiome studies, gut genomes, because for example, I work with birds, and other people have seen, oh yeah, those birds don't have any parasites, and we're finding out that some of these nice people that they have a lot of worms, and then you wonder what you're getting, right? Because the transcriptome or the genome, you don't really know what you're getting.
You know, that's actually a very good question in the sense that, and this is something that I'll be learning more, right? Because I'll be doing more microbiome work, starting next year. So that hasn't been my specialty has been more organismal, and this is something that has come up in conversation with my postdoc advisor with Donovan at UC Irvine about the pricklebacks. Is like, okay, which, sure that these genes are, from the organism or from the parasite or from the bacteria. And, of course, constructing phylogenetic trees, that we see that this common path that, okay, we see all these orthologs of that gene, and they're all fishes, and more than one. It's it's for the organism, or that gene corresponds to the organism. But there are some genes that, and I would have to talk to my postdoc advisor. There was maybe one or two that would be that there's still like a question in association with like digestive enzymes that we don't know if it's the organism or if it's the microbes, and so that's something that we're still teasing apart.
We have time for one more. Okay, I have
Okay, a couple of questions. How old are these fish in terms of like phylogenetics, and also what position do they have in a fish or marine fish phylogeny? Are they derived fish or sort of primitive fish?
So the estimates, so there's a fossil record for rockfishes, so that helps a lot. So it's about 8 million years ago for the rockfishes. And so, yeah, and then for some of those clades, I think it's around like 2 million years. So so with Pteropodus, so Pteropodus is a subgenus, so it's like around like 3 million years for that particular subclade, for that clade there. For the pricklebacks, I don't know if there's any good phylogenetic work aside from what we've done. We've used a couple of nuclear markers and mitochondrial, but nothing with calibrating with a fossil record. And there might be a fossil for pricklebacks, but I don't think there's anything available that, not to my knowledge.
Another question is, you talk a lot about sort of a consistent theme of your talk was multiple copies in all these genes that you saw, right? Was like the pattern. So that basically says there is a high diversity or genetic diversity in this group. Can you think of any reason why this group in particular will have more genetic diversity than others? Is there something related to something that you can associate it with?
We would like to associate it with like herbivory. When you look at teleos fishes, herbivory is not majority of fishes are carnivorous.
Right, the carnivore will be the plesiomorphic condition, right?
Yeah, and so, but just in fishes in general, you see a majority of them more carnivorous or maybe omnivorous. And what's interesting with the pricklebacks is that that they consume algae, and sure that there's some other I think the examples in the tropics with parrotfishes that they're scraping at the corals, and to what extent is it that the algae that they're consuming or the symbionts that are found there or these other organisms that reside there. But I guess it would be more interesting, or maybe we can lock it in that there's more copies if hopefully that we have these other genomes sequenced, right? And then if we only see less amount of copies represented, then maybe that the more copies would be associated with herbivory.
Thanks.
You're welcome.
Okay, we'll have the space for coffee and cookies.